Hello, my name is Jordan McKinney, and I'm currently a rising senior at Stanford University. Today, I'll be talking about my project on the neuromechanisms of juvenile aggression. So first, I'll be talking about the model species that I work with. For this project, I've used Ranitomeya imitator, also known as the mimic poison frog. These frogs display a number of characteristics that make them a good model species to work with. So first, these frogs are biparental and monogamous, and the tadpoles are reliant on the mother for an egg meal. They do this little begging display in order to display need for the mother. Looking across different species of frogs, the mimic poison frog tadpoles are a good candidate for aggression because variation in pool size leads to different displays of tadpole aggression. Because of smaller pool size and increased tadpole care by the parents, these tadpoles tend to be very aggressive with conspecifics, even cannibalistic in defense of their resources. What I want to try and do is determine the neural mechanisms for aggression that cause these displays. So additionally, some prior work that we've done is look across different brain regions and neural activation across those brain regions. So what we have found is that neural activation differs depending both on the brain region and the type of behavior, whether it be aggression or control. We looked at 4 brain regions in particular: the medial pallium, the preoptic area, the amygdala, and the ventral hypothalamus. And what we found is that neural activation is shown to be highest in the ventral hypothalamus and the medial pallium. So given this information, we were trying to really investigate the question this summer of what cell types influence aggression in juveniles. So we used this really cool technique called phosphoTRAP, and it's a technique for molecularly profiling active neurons in the brain during specific behaviors. And we used it to determine activated genes during aggression as compared to controls. So first you have a stimulus tadpole and the control tadpole behavioral assays. For the aggression behavioral assay, the tadpoles were placed with a smaller stimulus tadpole for 30 minutes and left in their tank. And then for the controlled tadpole is pretty much the same thing, except instead of a stimulus tadpole, they were placed with a small metal bolt. So these were recorded and behavioral tendencies were observed. The brains were then sectioned into 3 parts: the telencephalon, the diencephalon, and the spinal cord. Phosphorylated ribosomes, which are a marker for neural activation, were immunoprecipitated. And then this left activated neuron mRNA. RNA sequencing was then used to determine which genes in the brain are active during aggression. So through this, there are several interesting genes that are implicated in aggression, which are labeled in this chart. We have forkhead box protein 2, which is a transcription factor labeled involved in communication, and it is down-regulated. Proopiomelanocortin is a precursor polypeptide with multiple post-translational subunits, and it happens to be upregulated. Alpha MSH and beta endorphin are 2 of the subunits, and they're implicated in sex, aggression, and appetite. You have neurotrimin, is a down-regulated gene that's been implicated in aggressive traits in children with ADHD. You have this membrane protein, MLC1, which is upregulated, and it's been implicated in fly aggressive traits. Then over here, another subunit of POMC is ACTH, which is a hormone that stimulates the production of cortisol and mediates the body's stress response. GPM6A is downregulated, and it's been implicated in panic disorders. Then you have neuroserpin, also downregulated, and it's implicated in impaired explorative behavior. As you can see by looking at these plots right here, there are hundreds of genes that are up and down regulated that we got from this phosphoTRAP. We chose the ones that we've done prior research on, and we thought would be interesting candidates to look at for aggression in juveniles. Ultimately, we chose to focus on POMC because of its multiple post-translational subunits. So alpha MSH is implicated in feeding, aggression, and sexual behaviors. ACTH is a mediator of the adrenocortical stress response, and beta endorphins are endogenous opioid neuropeptides. So in order to confirm the effect of POMC on aggression, we used a technique called fluorescent in situ hybridization in order to selectively tag POMC neurons, and then we double labeled with a PS6 antibody, which is a marker for neural activation. So what this would tell us is that if we had a POMC neuron and a PS6 neuron that we could see in our images and they were co-localized, so they were right on top of each other, that told us that the POMC neuron was active. So you can see this in this image right here. We have DAPI, which is just allows us to see where we are in the brain. It stains tissue. PS6 cells are in green, so there's some right here. and POMC cells are going to be mostly around here. So through doing this, we actually saw that there's not a significant difference between POMC cells in aggressive and controlled tadpoles. So then we did these POMC manipulations. We used pharmacology to functionally determine the influence of POMC on aggression. So tadpoles were injected intraperitoneally with one of 4 drugs: alpha-MSH, which is an agonist; HSO14, which is a selective MC4R antagonist; ACTH, which is an agonist, as well as a vehicle that we used as a control. So these tadpoles were injected, they went through a recovery period, and then they were placed in aggressive encounters with conspecifics, very similar to this encounter over here. So through this, and through video analysis of the tadpoles' behavior, it was seen that tadpoles showed increased aggression with the HSO14 injections, as well as with the ACTH injections. So you can see that here. However, given that these injections did not produce any significant difference in behavior, there are a couple things I'm planning on doing in the future. So first, I'm planning on doing intracranial injections instead of intraperitoneal injections, as well as creating a dose response curve for the alpha-MSH and ACTH drugs. And this is in order to confirm these findings. I also want to manipulate the beta endorphin system pharmacologically, because that's another subunit of POMC that I think would be really interesting to look at in terms of aggression. And then in the future, I'm looking to do neural tracing experiments to determine receptor location, as well as projections to various brain areas. Thank you so much.